Hello, my dear students. Welcome back to Biology at Pujit. Have you ever wondered how a single fertilized egg transforms into a complete organism with a brain, heart, muscle, and organs? How does one tiny cell know where to form the nervous system and where to form the gut? Today, we are going to explore one of the most fascinating topics in developmental biology, or blastulation, then fate map, cell lineage, embryonic induction, primary organizer, Spemann-Mangold experiment, and Nieuwkoop Centre. And I think by the end of this class or this video, embryology will feel much easier and more interesting to you. Let's start. Blastulation. Blastulation is the developmental process by which the solid mass of cells formed after cleavage, called the morula, and it transforms into a hollow structure known as the blastula. The cavity formed inside is called blastocyst. It is an important stage because it establishes the first body axis. It prepares the embryo for gastrulation. It allows cell movement necessary for germ cell formation. General features of blastulation. During blastulation, cleavage divisions continue, cells become smaller and more numerous, a cavity or blastocyst develops, then blastomeres rearrange spatially. Polarity of the embryo becomes more evident. Formation of blastocyst. The blastocyst is formed by active transport of ions, especially sodium, into the embryo, osmotic entry of water, and separation of blastomeres. Functions of blastocyst: provides space for cell migration during gastrulation, prevent premature interaction between cells, helps maintain embryo shape, and distributes nutrients and signaling molecules. Types of blastula. There are four types. Coeloblastula, it's a hollow blastula with a large blastocyst seen in animals with moderate yolk, example Amphioxus, frog, etc. Stereoblastula, it is a solid blastula without a blastocyst. Blastomeres tightly packed. Examples include some mollusks and annelids. Discoblastula, where blastula formed as a disc of cells over yolk, seen in heavily yolked eggs. Example birds, reptiles, fishes. Blastocyst, which is found in mammals, is the blastula is called as blastocyst. Its components are trophoblast, which is consist of outer cell layer and it forms placenta. Then inner cell mass, it forms the embryo proper, and blastocyst, the fluid filled cavity. Significance of blastulation: it establishes embryonic polarity, prepares embryo for gastrulation, separates future embryonic tissue, and enables inductive interactions among cells. Fate map. A fate map is a diagram showing the future developmental destiny of different regions of an embryo at an early stage. It indicates which embryonic region gives rise to which tissue or organ. Fate map in amphibians. The amphibian fate map is commonly studied in frog embryos. Major regions: embryonic region or future fate. Animal pole, ectoderm, it will form the epidermis and nervous system. Marginal zone, mesoderm, it will form notochord and muscle and blood. Vegetal pole, endoderm, it will form the gut lining. Methods of fate mapping: vital dye method, cells stained with harmless dyes. Movement traced during development. Radioactive labeling, radioisotopes incorporated into cells. Fluorescent labeling, fluorescent markers are used. Genetic markers, GFP and molecular markers used in modern embryology. Importance of fate maps: helps understand organ formation, reveals cell movement during gastrulation, and it is useful in developmental genetics and regenerative biology. Cell lineage. Cell lineage, let's simplify the topic. It refers to the developmental history of a cell from the zygote to its differentiated descendants. It traces which cells arise from which parent cell. Characteristics: show sequence of cell divisions. It indicates timing of differentiation. It helps identify stem cell relationships. Types of cleavage related to cell lineage: determinate cleavage, it fixed early. A removal of blastomere causes defective embryo. Examples include nematode and tunicates. Indeterminate cleavage, the fate is not fixed early. Each blastomere can form complete embryo, examples vertebrates. Cell lineage in Caenorhabditis elegans, the nematode C. elegans has a completely mapped cell lineage. The features are adult has fixed number of somatic cells, every cell origin is known, widely used in developmental biology. Then Caenorhabditis elegans is an important model for studying cell lineage and apoptosis. Importance of cell lineage studies: understanding differentiation, stem cell biology, tissue regeneration, evolutionary developmental biology. Then we will go for embryonic induction. Embryonic induction is the process by which one group of cells influences the developmental fate of another group of cells. Here the influencing tissue is called inducer and the responding tissue is called responder. Types of embryonic induction: primary induction, first major inductive event, formation of nervous system by organizer. Secondary induction, it is the later interactions between tissues. Example include lens induction by optic cup. Homotypic induction, inducer forms same type of tissue. Heterotypic induction, inducer forms different tissue types. Mechanism of embryonic induction: steps, signal production by inducer cells, signal reception by responder cells, then activation of genes and cell differentiation. Important signaling molecules are BMP, which is responsible for epidermal differentiation, noggin for neural induction, cordin for BMP inhibition, FGF for mesoderm induction, and WNT for axis formation. Significance: organ formation, axis development, tissue differentiation, and morphogenesis. Primary organizer. Primary organizer is a region of the embryo capable of directing the development of surrounding tissues and establishing the body axis. In amphibians, it corresponds to dorsal lip of blastopore. It was discovered by Hans Spemann and Hilde Mangold. Organizer molecule. Important organizer secreted molecules are noggin, cordin, and follistatin. This inhibit BMP signaling and promote neural development. Functions of primary organizer: it induces neural tissue, establishes dorsal axis, organizes body pattern, and coordinates gastrulation movements. Types of organizers are: head organizer, they will form the forebrain induction; a trunk organizer, notochord and spinal cord induction; and tail organizer is responsible for posterior structure. So now we will study the Spemann and Mangold experiment, the classic transplantation experiment by Hans Spemann and Hilde Mangold in 1924 demonstrated the existence of the organizer. It is one of the most important experiments in developmental biology. Experimental procedure include the materials: early gastrular stage, amphibian embryos. Method: dorsal lip tissues from one embryo transplanted to ventral side of another embryo. Transplanted tissue integrated into host embryo. A secondary embryonic axis is developed. Observation: formation of secondary neural tube, notochord, somites, etc. Thus a partial twin embryo is formed. Conclusion: dorsal lip acts as an organizer. It induces surrounding cells to form neural structures. It demonstrated embryonic induction. Significance: foundation of experimental embryology, proved cell communication in development, lead to discovery of signaling molecules. Nieuwkoop Centre. The Nieuwkoop Centre is a group of dorsal vegetal cells in amphibian embryos that induces formation of the organizer, and it was named after Peter Nieuwkoop. Location: dorsal vegetal region of Blastula. It functions: it induces dorsal mesoderm, initiate organizer formation, establishes dorsal ventral axis. Mechanism: after fertilization, cytoplasmic rotation redistributes determinants, beta catenin accumulates dorsally, and vegetal cells produce signaling molecules. These are the important signaling molecules like WNT pathway, which is responsible for dorsal specification, beta catenin for organizer induction, and nodal proteins for mesoderm induction. There is a relationship between Nieuwkoop Centre and organizer. Organizer induces neural tissue located in vegetal cells. They are located in dorsal tip. Forms signaling center, they form organizing center. So this is the summary of embryonic development in amphibia. So if you enjoyed this lecture, this class, found it helpful, please don't forget to like the video, comment your doubts, share with your friends, and subscribe for more developmental biology and other zoology lectures. See you in the next class. Thank you.